discussing sustainability from a multidisciplinary perspective, for, integrating all fields of knowledge in this effort towards a more sustainable planet. So today, that's exactly what we are going to talk about. The theme of our webinar today is sustainability in the field, integrating biological resistance mechanisms towards balancing agriculture and the conservation of ecosystem services. So again, it's an honor to have you all here today. It's really great. It means a lot to us and to this university as well, as we begin to these interesting partnerships, and we can gather people from all around the world. We have lots of students from India in the chat today as well. So I'm seeing here lots of professors and researchers from the UNIVASF. So yeah, you are all very, very much welcome. Just about the structure of this webinar today, we have 3 speakers. Each speaker will have up to 30 minutes for their individual presentations. Of course, they can choose to either use the whole 30 minutes or just a part of it. After each presentation, we have decided before, we will have in-between questions. So after each speaker, the audience can raise their hands here and I can organize their questions. So you will be able to interact directly with the participants, okay? And after each presentation, we'll have a question and answer session. And okay, I think that's all for now. So thank you again. And I would like to begin by inviting Dr. Monica to start her presentation. And I will be here should you have any questions.
Okay. Yeah. Well, just a question in the beginning. Am I audible to everyone?
For me, it's okay.
Vlad, am I audible? Okay, okay, yeah, good. Yes. Well, good evening. Hello. It's evening in India over here. So my international students, those who have joined the webinars, a formal greet word when we welcome someone to our board, we say namaste. So it's like that. I'll begin with. I would like to request Vlad that whenever I ask you to change my slide, so kindly help me with that. Presently, I'm turning off my camera so that there should be no, any issues in between. Yes, thank you, Vladimir. Thank you, University, for considering me as one of the speakers in the most emerging, rather I would say the hot topic of the research, sustainability in the field. So can you just change my slide?
Okay.
Yes. Students and researchers, I hope nowadays all around the world, we are coming aware of the very common term, climate change, and people are like, right now, I was just having a talk with my co-speakers. I was mentioning it's like around 25 degrees centigrade in India right now at my place. And over their place, it's like quite heat up. So there are little bit shifts always, but we need to be aware of that what do you mean by climate change? As you can see in my slide, there is a dome shape which mentions climate risk spectrum. You can see when we say a climate risk spectrum, these all factors comes into mind. For example, flooding, drought, extreme rainfall, heat wave, increasing temperatures, loss of biodiversity, and many more. So today on this platform, I'm going to focus upon one of the topic, which is the salinization, which is the salinity stress. Okay, next. Next, please. Yeah. This slide is very basic for the learners, for the listeners, for the experienced people, just to let you know that what is the difference between abiotic stress and biotic stress, because both these combinations becomes the part of the climate change. If you will click Google, they will say that climate change means something, variations in temperature or some other kind of things. But I would like to point out over here that climate change does also include the biotic component well in detail. So this is just general information for all those students and researchers that what is abiotic? Something which is physical, biotic something biological. So all these stress factor the plant is always surrounded with. So the plant is quite smart. Plant respond to the stress conditions, howsoever the climatic conditions are. Yeah, next please. Okay. Uh, all of you can see that I have shown the depiction of the lands being affected by salt stress. Here I'll take a little pause and I would like to brief you all about that why I have chosen the salt stress for my studies. If you can see from a national point of view, the whole map of India, you can see a lot of land is already impacted with salinity stress. Someone might question that, ma'am, or like, what do you mean by salt stress? So salt stresses means when there is a difference in the concentration of sodium and chloride ions, when there is an uptake and leakage of sodium chloride ions, that becomes the salinity stress. But why it is very much vulnerable to the plants? Because if salt stress is happening, then osmotic stress is coming, then ionic stress is coming, then nutritional imbalance is coming. So due to that, there is a change in the entire metabolic system of the plant. There is a change in the growth and development of the plant. There is a change in the redox homeostasis of the plant. So entire system of the plant get disturbed. So that is why I have took up salt stress. And yes, as I am working in Maharashtra, I am working in the college around Pune. Here most of the lands fall in the categories of the coastal areas where the land is heavily impacted with the salt stress. That is the other reason why I have chosen for the salt stress studies. Coming back to the slide, the present slide that you're observing, left hand side is the global distribution. So I think Europe, North America, Mexico, South America, Africa, Australia, almost all continents are heavily impacted with the salt stress, and which is, yes, is on the alarming rate. Yeah, next. Yes. Yeah, I was just talking you about that. What do you mean by salt stress? This is a very simple diagram. I have given the reference below. I have given the hyperlink below. Those who are new to the studies can click this and can have an idea later on my presentation I can share. So you can see that there is a plant and underground you can see certain molecules. Okay, so like potassium ions are there. You can see how the plant is responding. There is an uptake. As a result, if there is a salt stress, there is a decrease in chlorophyll content. There is mishappening at the level of the leaf. You can see stomatal closure. Of course, those who are the basic biologists, this is very basic, fundamental thing. If the plant get salt stress, then of course there is a closure of stomata. On right hand side of this diagram, once this these are happening, parallel to it, what is happening is the production of reactive oxygen species. ROS stands for reactive oxygen species. Now, what do you mean by reactive oxygen species? In general, when a plant encounters stress, be it abiotic or biotic, as a natural defensive mechanism, plant respond and there is an activity of the redox homeostasis, so which is the production of reactive oxygen species. Okay, this is what I have told you. Next please. Next please. Yeah. Okay. Here I would like to tell you that the data which I am presenting, the entire work is done by one of my student. She was working as a third year student, BSc Biotechnology. Her name is Sapna Sharma. So in her BSc Biotechnology, I have given her the project to check the impact of salinity stress on spinach. Now, why spinach? Why? Because whenever we start with any studies, being a plant scientist, we always get stuck with the plant material. So why spinach? Because it is heavily grown again in my region, and it's a leafy vegetable. And it's a very common understanding that if it is a leafy vegetable, it is more susceptible to the stress, and it is more susceptible to the uptake of ions because it's completely a leafy vegetable. So entire leaf is available to get impact with the stress. So that is the reason. Second, it is one of the staple diet during the winter season all over India. So, and third thing, very cheaply available in the Indian market, we can eat and we make a lot of things. So my focus is on like if the spinach is eaten a lot, because there are a lot of reports on the leafy vegetables getting impacted with many kind of stress. So I have given her that kind of study, that let us study some of the physiological and biochemical parameters about the salt stress. So what is the methodology behind is that the seeds were germinated. Okay, and the second thing, I would like to tell you all that I have used the hydroponics rather than the soil media. If anyone do not know about the hydroponics, please let me know after my presentation, because that is one of the way, and rather one of the upcoming entrepreneurship setup, growing plants in the hydroponic way. Hydroponic way simply says that it is a soilless thing. We give some nutrients to the plant in the form of a liquid, and then we give the stress. As a student, one thing can come into our mind that, ma'am, why water? Why you're not given the stress in the soil? So again, a very common answer could be that ions can be, easily approachable to the plants to cause stress through the water media rather than the solid media. And apart from that, some of my students were interested to learn this technique of hydroponics so that they can set up their own entrepreneurship. So I have told that let us have the hydroponics. So the seeds were purchased from the market, from the good grocery stores. They were the certified seeds. We make it sure that the thumb rule of the plant material is that it should be 100% germination index should be shown. So the seeds were germinated for like 3 to 4 days, and of course in the plant growth chamber, the humidity was maintained. The 25 degrees centigrade temperature was there. Then we have allowed to grow the plants till 7 days, till 3 leaf stage, and then we have given the salt stress. Okay, so the question might arise that what stress was given at what concentration. So I have just considering the time constraint of the project, I have given two concentrations. So one is control, another is a 100 millimolar. So I opted to give that concentration, and then the students went ahead for the practicals. So once the salt stress was given, we have opted two parameter for studies: morphological, physiological, and as well as some biochemical parameters, because we cannot study the entire thing at the BSc level. So next. Yeah, so the results were absolutely I have thought of. If you can see on the right hand side, I'll just take time to explain some of the results. X-axis shows the salt concentration. Okay, if you can see, I have taken four different concentration: very low 25 millimolar till 100 millimolar, because usually the salt stress beyond 200 millimolar, if you'll go, then the plant will die. So remember, here I'll give you a tip to all of the researchers and the students, that if you want to give stress, give the stress up to the sublethal level, so that at least plant should respond. You should not give the stress beyond the concentration where the plant just die away. Okay, so screening is really important. So I have, yes, always a control should be there to check that whether you're doing your experiment in the right way or not. This is all learning phases. You can clearly that orange bar shows you what root length, blue bar shows you shoot length. If you can see, with the increasing concentration of salt stress, right, from 25 till 100, and I have taken only a gap of 25, 25, and 25. You can see how heavily the spinach, it's get impacted. Actually, it has started impacting at the concentration of 50 millimolar. You can see a big difference. So yes, you were right in selecting the right concentration. That is a satisfaction thing for us. Second thing, as you all know, that spinach is entirely a leafy vegetable. So there was one of the morphological feature that I told to my student that please observe what is happening to the leaves. So what she told me, ma'am, the leaves started degrading. There was change in its visual characteristic. There was change in leaf textures and turgidity, and of course leaf curling or rolling as well. So we were quite disturbed, because 100 millimolar is already alarming in the Pune region. So, and this is heavily consumed. So let's see what I'll do with this kind of research. Yeah, next. Okay. Yeah, previous one, please. Okay, this is all. Okay. So, okay, fine. So I would like to tell over here that when plant encounters stress, as I mentioned you, that there is a production of reactive oxygen species. So few examples of reactive oxygen species are hydrogen peroxide, then there are superoxide radicals, kind of some other as well. Amongst them, hydrogen peroxide, when this is released, then catalase enzyme comes into function. Okay, that is called oxidative stress. So what I want to tell you, that when the plant had sensed that some salt is coming into it, when there is an uptake of salt of different concentration, the catalase activity should get proportionally increased as a defensive mechanism. Now what catalase enzyme does, it it converts that hydrogen peroxide to that particular radicals which are good for the plants. Okay, what I've written in the red part is catalase. In short form, we call it CAT. This is internationally renowned that yes, you have to depict catalase in the form of CAT. So the CAT absorbance with respect to stress assumed an incremental graph within the 24 hours and mid of 48 hours, after which it was characterized with a sharp downward decline. Here I would actually, I forgot to mention that the entire experiment was set up till 72 hours. That is another thing which is important while working with the stress experiments. There are 2 or 3 thumb rules. What is your plant material, what is the concentration of stress you are giving, and what duration you are giving, because all these factors are correlated. If suppose I would have went ahead of 72, say 96 hours, then I would have not get any data. By that time all kind of post transcriptional or post translational things would have elapsed. So it is ideally the suggested durations are up to 72 hours. That is suggestible because during that time the plant respond. Okay, so you can see in this graph clearly that with the increasing concentration, there's an increase in catalase activity. Yeah. Apart from that, there are many other actually assays we can do. Catalase is there, then lipid peroxidation is there, which you can see in many other. You can do that kind of literature survey as well. Yeah. Next. Okay, so a very basic experiment I have given, chlorophyll, and the next slide with the protein estimation. Uh, if you people are the biologist or biotechnologist, right from your second year or third year of your undergraduates, you must be having the experiments where you have to measure the chlorophyll content and also the protein content. And I always used to wonder when I take practicals of masters and even when I was pursuing my PhD, even in my PhD, I have done chlorophyll estimation. And I was used to wonder, oh my God, this is such a small experiment. Why? Why chlorophyll estimation? The reason is the photosynthesis. The reason is the photosynthesis. When stress happened, all these reactive oxygen species, they impact the photosynthetic machinery of the plant. And if photosynthesis is not happening, then of course we will have no producers and we cannot survive. Henceforth, it is a mandate if you are doing some experiment related to stress and if you are looking ahead for a good publication, you should do chlorophyll estimation, especially if you're dealing with the leafy vegetables. Okay. So, and my students are also very much easy to handle such experiments, and I want them to do on their own. And yes, of course, see, if you can see, the chlorophyll test was performed for these 24 and 48 hours and results in the increment in absorbance with an increase in salt concentration. So you can see again, there was a control 24 hours, 48 hours, 72 hours, and the highest concentration you can see. And then there's a sharp decline because beyond that, something else is happening, which is a question to all of you, that why is the decline happening? Okay, yeah, next. Okay, as I said, protein estimation, Lowry method they have used, and of course, at the concentration of 100 millimolar, and the results are there. It's directly proportional to the increasing concentration of the salt stress. Yeah, next. So, then we were having a brief discussion that, ma'am, what we should conclude. Then we come up to the 4 key results of that small experiments, which was done within the 3-month of span. So what we can clearly say that there's a decline in both root and shoot growth as salinity stress intensified. Second, leaves exhibited pronounced yellowing, indicated severe stress conditions. Third, chlorophyll and catalase levels initially showing an upward, but then of course there's a sharp prolonged exposure to salt, and then probably the plant would have started dying. And interestingly, protein content exhibited a distinct pattern, displaying a steady increase characterized by consistent increments over time. And this study has not been done before. It's an honor for me that my students are doing such a good research in their own. So yeah, next. Okay, so what to do? Because people can't stop eating spinach. It's a healthy vegetable. It's a leafy vegetables, globally consumed. Even when you fall ill, the patients, they are given the spinach soup. And if it is so much prone to salinity stress, which I've already shown in the initial of my slide, that globally as well as nationally, the land is heavily impacted with the soil stress. What you have to do is you have to enhance soil management practices. Now, what is that? You have to improve the drainage systems. You have to do certain kind of crop rotations. Second, of course, the world is moving ahead of genetic engineering. Now there is a concept of genome editing. So yes, we can have genetic engineering crop as well. Then we should have sustainable irrigation practices. And of course, fourth, ecosystem based approaches. Yeah, next. Yeah, I am just about to conclude my slide. I would like to give acknowledgement to our principal, respected principal, Dr. Sanjay Kharat, Modern College GK Pune, head of the department, Dr. Vinay Kumar, Mr. Vladimir Nunes, Brazil, who had allowed me to speak to present my very, yeah, one of the favorite area, which is the stress biology. And let me tell you, Vlad, that it just happened, the coincidence that my student just submitted this project report in the third year, and we got very good results, and she learned a lot from the hydroponics experiments. And the photo which you can see is of her only. So, and she was also appreciated during her presentation. I hope I can do something good for her. And of course, ARI International Relations Department, I have to acknowledge. Have to acknowledge Department of Biotechnology and Department of Science and Technology, Government of India, because all the facilities which have been provided at my department, because our department is supported under all kind of government organization, whether UGC, DBT, DST, you say BARC. We have very good facilities, more than 10 crores projects we are worth money of project only Department of Biotechnology is having. So that is acknowledgement for all of them. Yeah, next. Yeah, thank you for patience listening, and be careful while consuming spinach. Wash it thoroughly, and do not overcook, because again you are taking a lot of salt inside you. Yeah, thank you for your patience listening. Yeah, Vlad, over to you.
Okay, thank you very, very much, Dr. Monica, for this extraordinary presentation. I really enjoyed it.
Yeah,
And
thank you.
before I open to questions of the audience, I do have some comments and questions of my own.
Okay.
This is very interesting because, our universities here is located in a semi-arid region,
Yes.
the semi-arid region of Brazil. We have a very
Yes.
characteristic ecosystem that is called the Caatinga.
Yes.
It's a
Yes.
semi-arid ecosystem in Brazil.
Correct.
And we suffer a lot from water stress, water deficit, lack a lot of
Yes.
water.
Yes.
And as
Yes.
a result, we also suffer a lot with salt stress.
Correct.
And I saw your mention to soil management practices and different agricultural practices, such as agroforestry and integrated management systems, right? And that's very interesting because we also explore these things here. Agroforestry systems are very widely promoted in this region as an
Yes.
alternative to the monoculture system of farming.
Yes.
This region here, where we are located, is also one of the greatest areas in Brazil where we have fruit farming, irrigated fruit farming, and
Okay.
so and of course, it's monoculture. It's meant for export. It's not necessarily concerned with soil and nature and
Yeah.
so that's a very interesting input you bring here. And I do have
Okay.
a question. Let me just share your presentation
Yeah.
again so I can.
Yeah. No issues, please.
Right. Um,
Vladimir, your voice is breaking.
Yeah, your
Oh,
voice is breaking. And
sorry.
yeah,
It's back.
yes.
Okay, here. Um, it's in, it was here, I guess. Yes, it was here.
Yeah.
Okay, so you showed here your experiment, and we have different salt concentrations to which the spinach plants were exposed, right? Uh,
Yes.
we have a test, a control concentration here, and we have the other increasing concentration. So the region where you are, where this crop is usually grown, what is the salt concentration that is usually found in the soils of the region?
Okay. So, good question. And if you're begin to with the stress studies, you should know from where you have, got the seeds. And Vlad, I have to tell few things actually. Ideally speaking, all over the world, the most heavy concentrations is found around 200 to 500 millimolar. Actually, plant will die, beyond 500 millimolar. Even the plants, I mean, the salt tolerant, its range is 200 to 500. So over here, most of the salt, what we have, check in our laboratory after a lot of screening,
Yes.
because we have actually started from 10 millimolar till 25 and many more. So we have found 100 to 150 millimolar in our region. So and Vlad, if you will go on the Google Scholar, most of the people till date, they are taking last concentration as 100 millimolar. As I told you that while working with the stress studies, you give only that much of stress that much plant can bear.
Yes.
After that, the plant will slowly and slowly start dying only. You will not get any of the studies. So that's what you've seen. There is a very little difference between the control and 25 millimolar concentration. The moment I have doubled the concentration of the stress, plant started responding.
Hmm.
And for your additional information, the experiment was set up in 3 replicates and was repeated twice to make sure, because these are third year students, so I have to be a little bit, I have to look back whether the experiments are doing in an ethical way or not. So these things are also kept in mind. So I would suggest, if you're working with the salinity stress, don't go beyond 150. 100 is enough. 100 millimolar is enough. You will get good results even if you're looking for the molecular studies, or you're going for epigenetics, or you're going for post transcriptional microRNA, post translational. 100 millimolar is like okay. And yes, within 72 hours.
Hmm.
So this is the thing.
Okay. Excellent.
Yeah.
Excellent. Thank you very much.
Yeah. Yes.
All right. Does the audience have any questions you would like to pose to Dr. Monica? Your
And okay, yeah, Vlad, I
oh,
want to tell you one more thing, and
okay.
to others also. Why certified seeds? If you want to do a clear and you know good research, try to take the F1 progeny. You have to make sure when you are going to the field, that is the F1, because if it is F2 or F3, then that's become the stress memory.
Mhm.
In stress biology, there is one common term called as stress memory, and there are types of stress memory: somatic and trans. So if the stress memory is not inherited by the till F3 generation, you might not see such good results. So you have to make sure that certified seeds are actually certified. They are not like bought from any grocery stores. And I actually make sure that the seeds should be certified from the authorized organization. I mean, it should not be like genetically engineered. It should not be a hybrid, which is a Herculean task. Let me tell you, because no disclosures are there.
What is the origin of the seeds you use in your experiments? You have a certified agency that provides these
So
seeds?
yeah, so Vlad, I have done my PhD from National Bureau of Plant Genetic Resources, which comes under Indian Council of Agricultural Research, which is the number one in the global Indian university. So we have seed bank facility over there at my bureau. You can type Google NBPGR New Delhi. You will see that gene bank is there, seed bank is there. So there is an agreement. We send that agreement, and they make sure that the seeds are 100% germinated and they are pure, whatever you're expecting. So you can see over there that passport data is also there. Everything you can do it online. So they maintain that seed bank. So that's what. Yeah.
Interesting.
Yeah.
That's great. Thank you.
Yes.
All right. All right. The audience can pose any questions if they wish. I have no questions. All right. I think somebody is writing on the chat. So somebody asked the question, Monica, in the chat. I'll read it to you because it's written here. The question is, why does the high salt concentration in some soils happen naturally? Is it possible to make these areas is it possible to make these highly affected areas productive again? Good question.
Natural. We cannot fight with the nature, you should actually know that what is stress, and when does salt become stress. So so one of the phenomenon is the weathering of rocks. Some of these geographical phenomena are there that releases salts into the soil. And then the second reason is the natural geographic location of the plant from where you have taken up. Overall globe, they have different kind of salt concentration naturally, which of course we can't avoid. So first thing is that only, yes, we can try to escape from the salt stress by, as I mentioned in my last slide, by handling some of the ecosystem practices. And if genome editing turns to be a cheap technique, then we might have everything resistant to salt stress. Might be someday. Which of course should not happen. Balance is there, should be the nature. Something should be sensitive, something should be resistant, then only you can have mixed progenies.
Mm-hmm.
So yeah, thank you.
All right. Do we have any other questions? So I think for the moment we have no other questions. So thank you, Monica, again for your presentation and for being available. It's been an honor to have you here.
Thank you.
All right, and I hope you can manage to come here in August.
Yes, I'm trying.
Yeah,
I'm trying. I'm working on the abstract actually. So let's see.
Okay. Just so we know, the audience knows, we are organizing an international congress on climate change and its consequences on semi-arid regions, which will occur here at the university. So we have managed to bring some researchers from all around the world. We hope Monica can join us. We are bringing a friend of ours from India as well, who is a professor at the IGNTU. And that will be in August. So you will be informed in proper time.
Yeah.
So, sorry? Oh, okay.
Yeah, nothing. Carry on, please.
All right. Thank you, Monica. So I think next is Dr. Yana, right? Okay. Can you share your presentation, or is that okay?
Yeah, I tried. So just to let you know, I'm not a doctor yet. I'm still in my PhD. So
okay, it's almost.
I just want to correct that.
All right.
But soon, right, Jana?
Huh? Soon, hopefully. So you see my screen, right?
We do.
Okay. Yeah, so as I already said, I'm a PhD student from Bielefeld, Germany, and today I would like to introduce you to the topic biological pest control in agriculture. So this was the main topic of my PhD. And yeah, but first of all, it's not working. Did this slide change for you?
Yes, Jana, I did.
Okay. Yeah, first of all, I would like to just define a bit of biological pest control. So the definition is that it's a protection against pests and pathogens, and it's sourced from nature. There are several advantages that normally there's an existing degradation pathway, and the biocontrol agent is highly specific to their target organism. And normally it should cause minimal negative effects on other organisms, so on non-target organisms and the ecosystem. And for sure, there are some requirements. So it should be non-pathogenic to humans, and it should at least be as effective as chemical control. And this biocontrol agent need an existing ecosystem mechanism to balance their population. That is important because I would like to show you some examples. And the first one are aphids. So they are feeding on plants and sucking their phloem. And there was so ladybugs are used to control them. The problem there was that people decide to use in Europe an Asian version of the ladybug, and this is now invasive to Europe. So the European ladybug is not, so it's still existing, but it's getting lesser. And a second example is the European corn borer. So the caterpillar is feeding on plant tissue, and the adult version is feeding on nectar. And this can be controlled by trichogramma. And trichogramma is a polyphagous wasp that is endophytic to insect eggs, for example, to this corn borer. And a third example are beetles. So their larvae are feeding on roots on several plants. This species is feeding on potatoes, and they can be controlled, or they are tried to be controlled by Metarhizium bruneum. That's it's a fungus. And what I did in my PhD study was I used the intercrop Phacelia. I would like to go into a bit more detail for that, because otherwise it's hard to understand the whole topic. So Phacelia is an intercrop. That means it is planted between main crops. So for example, as you see on the right side, so there are several modules you can use, and this is always a change of the fruit to vary your plants on the field. And Phacelia is, for example, here started in the third module for green manure. And the potato, for example, is planted afterwards. And these are also, so I'm also working on potato in my project. And Phacelia has a variety of advantages. So it's soil loosening, it's a really good source of food for insects, especially for bees. It's crop rotation neutral. That means that it has nearly no own pests that can grow then in the field. It also has nutrient fixation effects, and it suppresses weeds. Okay, so the second organism I'm working with was Puchonia chlamydosporia. It's a nematophagous fungus. And as you can see on the right, it is growing in hyphae, and these hyphae are then connected to the eggs, and they are just kind of feeding the eggshell of nematodes. And this fungus is kind of special because it's also growing endophytically in plants, in a variety of plants, but it has no parasitic effects. It's just neutral or has positive effects for the plant. Okay. The third organism now is the nematode. Melodygyna hapla, so it's a plant parasitic nematode. That means that it relies on plant roots to grow in there. And you have an exophytic phase, so this is really the eggs and the worm itself. And these small worms then enter the roots, and they are growing like these giant cells in the roots, and they are feeding from the plant. And this causes a lot of damage in important crops for food, for example, carrots and salad and potato. So you see they are kind of deformed. They are building these giant cells, also called galls, and they lead to less water supply for the plant, for example. And potatoes look like this if they are heavily infected by Melodygyna hapla. Yes, and to make the story round, we need the important plant for the human being. So in this case, it was the potato plant as human food source. So this plant parasitic nematode is feeding on the potatoes, while the fungus is feeding on the nematode. And to control the nematode, the fungus is added to the seed, but not to the seed of the main crop potato, but to this Phacelia plant. And if this plant with the fungus is then sown in July or August, the fungus can grow in soil, can feed on nematodes, can grow endophytically in the plant, so can establish itself in the system. And then in the next year, in March or April, when potato plants are planted, then the fungus is feeding on the nematode. This arrow is kind of switched here. And then, if the fungus then feed on the nematode cannot feed anymore on the plant. So we want to protect the potato plant from nematode feeding sites. So that's why I did some greenhouse experiments at first. So we had our Phacelia plant and inoculated them with Melodygyna hapla eggs, and with the fungus in three different treatments. So there was a control treatment without the fungus. There was a treatment with just a spore suspension. So this fungus is growing also by liquid fermentation, and if you use the spores, then you can also inoculate them just as a liquid. And additionally, I did this seed coating, so I added the fungus to the seed. I would like to go into some detail for the coating because it took me like 1.5 years of my PhD, and it was a lot of work. So what you see here are them pictures. So this is the untreated seed, so it's quite porous. It has it's not a really smooth surface, and after the coating it looks like this. So this kind of rough, and it's a lot of surface. So the surface increased as well, and I looked into more detail. So you can see here the structures from flour and the gelling agent I used for this coating, and I also used potato starch for this coating, but you can also see this fine, really small fungal spores. Okay, back to the experiment. After the inoculation of the soil and the growing period of around 3 months, we took from all of the treatments and put a tomato plant onto that soil. So we use tomato in this case because it's easier to grow in the greenhouse, and Melodygyna, so the nematode, is nearly as good growing on tomato than on potato plants. And after 50 days, we calculated a gall index. I will show you in a second what that means. So what we did is we counted the number of eggs per pot. And what you can see here, so the dotted line indicates the inoculated eggs at the beginning of the whole experiment. And in the control, they really increased, and they are around 10,000, for example. And with the spore suspension, we could decrease the number of eggs per pot by 75.5%. And with the seed coating, we could even more decrease the number up to 95.6%. And on the left now you see the number of parasitized eggs by the fungus. You can see that microscopically. So we just looked at the eggs and see if they are parasitized or not. And in the control, there were around 15% parasitized. That's normal because under the microscope you can see a broken eggshell, for example, and that is an indicator for parasitism. But a broken eggshell also can occur when the egg is was broken before or was broken during the extraction, for example. In the spore suspension, there were around 20% parasitized, and in the treatment, in the seed coating treatment, there were up to 25% parasitized. Okay, so now back to this gall index on the tomato plant. So on the left, you see this is, so the index is from 0 to 10, and 2 is like, okay, there are some galls, but it's still fine for the plant. A gall index 4 is, okay, there are a lot of galls, but plant can still survive. And 6 is there are heavily galls, and there are also galls on the main roots and not only on the fine roots. And yeah, we can see that the seed coating has a gall index from around between 1 and 0 and 3. And in the reference group, so in the control group, the gall index was up to 5. And yeah, this is just again, so if the number of parasitized eggs increase, then the gall index will decrease. Okay, so what are the advantages of biological pest control with seed coatings? First of all, in this case, if you use an intercrop plant that is also fixing nutrients, and then you can add a second organism to that, you have like both in one step. Additionally, higher main crop yields per hectare can lead to less farmland. So if you have like a higher yield on a smaller field, then you can use the farmland for yeah, as another option. So if it would be optimal if it's not then used for building houses, but it's just then for forests or free grassland or whatever. And another point that is an advantage for the seed coating is that farmers don't need extra water because they can just bring it out with the seeds, what they do anyway. And you do not have to resolve something in water to bring it out. And that also caused a reduced time for farmers. Still, of course, it's all good ideas and there are a lot of opportunities, but registration processes are hard and expensive. And yeah, there is a lot of paperwork and a lot of research to do before you can register such a product. Additionally, so a biocontrol agent is an organism. So if the circumstances like the abiotic parameters that Monica already talked about are not optimal, then they cannot grow in the field. And it's at the moment it's more expensive compared to chemical control because you have to cultivate the microorganisms and so on. Furthermore, the products that the customers can buy then are at the moment more expensive because the whole process is more expensive. And that's why definitely more research is needed. And yeah, that's the end of my presentation, and I would like to discuss with you this topic.
Thank you, Jana. Thank you very much for this. This is very interesting, and it's amazing because this whole topic we are all discussing here, and people who are from my region here as well, they will relate to this topic because it's a very, very common background. Before I open again to everybody, I have some comments and questions. That's interesting. I mean, everything you just spoke. We saw here that there are ways of there are alternatives, biological alternatives to the use, for example, of agrochemicals, right? From chemical pest control, and even in significant crops like potato, which is a significant crop worldwide. And I was wondering, so we have these alternatives to chemical treatment, and of course, because there are concerns about the very use of chemical control in agriculture because it has consequences, right? Unfortunately, Brazil is not an example of a hard stance against chemical control, because chemical control here is a very widespread practice, and that's very shameful. And we are all here very aware of the dangers associated with it and the need for biological control for these problems. And before I comment also about the higher cost of these biological control and the final product, what is the stance in Germany with regard to chemical control? I was wondering about that.
Yeah. So so there are a lot of strict rules in Germany for chemical control, especially for nematodes. So there are no chemical products allowed in Germany at all against nematodes. So there's no way to use chemicals. And for other insects, it's also for insects and even for weeds, it's kind of hard. There are like restricted areas. So there are I have no English word for that. So there are areas where the government decided to protect nature, so nature as far as nature. And in these areas and next to these areas is still land that is used by agriculture, but they are not allowed to use any kind of chemical treatments for anything. So it's in Germany, it's quite strict and there are a lot, but still so there are some biological products in Germany at the market, and I also know that is at least one is in Brazilian because I figured that out. It's also using Puchonia as this fungus I was talking about as an biological agent. So and it's quite famous. So you can check that online. It's called Rizotec, and you can find a lot of studies about that, and they are mainly from Brazil.
Okay, interesting. I didn't know that. If you could write that on the chat, it would be great for me to,
sure.
to share with the participants as well. And Monica, I think Monica has a question.
Yeah, Jana, yeah, very good presentation.
Thank you.
Yeah, so this is called biotic stress, right? Yeah, kind of.
Yeah.
Yeah, Jana, actually, I want to start working with the biotic stress as well.
Yeah.
One thing you can take it as a question or as a comment. I think working with the biotic stress is a challenge because that is season specific, right?
Yeah, totally agree.
So you have to wait for the plant and you have to be very much friendly with your plant that when it is going to grow and which stage you have to work upon, right?
Yeah.
So Jana, suppose I have started working with some viral pathogens, okay.
Mm-hmm.
And if the season is not there, if the season is not there of the crop being infected with that virus, then how can I give artificial virus stress to that plant? How can I give?
Yeah, that's it's not easy. So I also conducted, so the study I presented was one of the ones that worked best, to be honest. So I did a lot of other studies, and if the organisms are just not growing, you can't do anything about that. So if
Correct.
if the nematode population, for example, is just
Yes.
kind of overpopulated, so or it's just old or it was stored for too long time period, so then you'd have just no chance. If it's not growing, then it's not growing. That's the point. Yeah.
Okay, John. Thank you. Yeah.
Okay, does anybody else have any questions from the audience? Come on, guys, don't be shy. I know it's a lot of pressure to put the participants in front of the speakers like this. I also had a comment about what you told, that it's one thing that we develop these mechanisms, and it's another thing that they are adopted widespread because it's a new technology, and because the costs associated with the implementation and also with the final product. We face a similar thing here, but with a far less sophisticated thing, that is organic production. I mean, we have the encouragement for organic production here, without using agrochemicals, and with or without using biological pest control. Of course, sometimes people use that even without, a lot of support, institutional support or financial support. But common complaint in the market is about the price of the final product. So yeah, they complain that the final product is more expensive than the other ones. And so it ends up not being that much accessible. But that is a greater issue, right? Because it's cheaper for the status quo to maintain the use of chemical control, because it's cheaper. They don't have to make any effort to change the reality. So
yeah, that's true.
Yeah, so it's, I mean, whether people are developing cancer 20, 30 years from now because of what we are eating right now, it's not a problem. Governments often care in many places. That's certainly the case in Brazil. And Brazilian colleagues who are here know that I'm not just talking nonsense here. We all know that current regulations in Brazil are wrong, and we have a lot of problems associated with that. Monica has another question.
Yeah, Jana, sorry to ask you, but that is my curiosity. So, Jana, why you have taken up tomato plant? I think I've seen that photo of tomato, and because it's close to potato.
Yeah, so first reason was it's close to potato, and second and main reason was it's easier to cultivate in greenhouse. So if you want to cultivate potato in greenhouse, it takes like forever. So and tomatoes are growing like quite fast, and you can culture them easily. And yeah, so for the nematodes, we tested that before. So for the nematodes, the development was the same in tomato and potato, and that's why we decided for tomato. It's just like a better. So it was for us easier because of the time schedule.
Okay, so are you expecting to develop some kind of formulation to, as in like to transfer lab to lab to field transfer kind of technology you're going to develop, like kind of formulations?
We already developed, so it was in cooperation with a company from Germany. They are normally just selling seeds, so like a variety of seeds, and they have a already existing system for seed coatings. And they already added Puchonia, so this fungus, to their seed coating. And we also tried that in greenhouse, and we also tried that in field. But unfortunately, there was like a really big flood, and our field experiment was just gone after 2 days. So there was no chance to repeat it. But yeah, so we already tried to upscale this whole process. But yeah, as I already said, it's also for the company, it's quite expensive, because just to cultivate the microorganism is quite expensive. That's the most critical point about that. But it was working in greenhouse, so in greenhouse it was fine. But yeah, field season was then gone with the flood, so no chance.
That's okay. That is a part of PhD. Yeah. Okay. No more questions. Yeah.
All right. So does the audience have any questions now? We have a question from a participant in the chat, Larissa, my friend Larissa. She says it's a very good presentation. I would like to know if this method could have also been used in an agroforestry system.
I'm not sure what is meant with this system, to be honest. Maybe I just don't understand the word. So
I don't want to interfere, but maybe they mean if it was or could be done in field experiments and not in a greenhouse study, for example. But not sure.
No, an agroforestry system is a different a different planting system that does not employ. It's meant for organic cultivation, does not use chemical treatment, and it's self-sustainable. So we use different crops like fruit crops and other kind of species to maintain a system, a self-sustainable system. Maybe
Okay.
Monica would have an input about that because she mentioned agroforestry systems in her presentation as well.
Yeah, so maybe I go first. The seed coating I developed is completely be usable for any kind of bringing it out to the field. So it's starch. This is fine for every organism in the field. It's even a food source for a lot of organisms. This gelling agent is a biopolymer, so it's produced by bacteria. It can be metabolized by the fungus, for example. That's where we use it. And the rest, so this rock floor is already a natural product, so it's completely biodegradable, I would say, this coating. That was one of my requirements for the PhD, because otherwise you wouldn't be allowed to bring it out in Germany.
So yes, I guess the answer is definitely yes.
Yeah.
It wouldn't be a problem. All right. Does anyone have any other questions? So I think for now we have no questions. Thank you, Jana, for your presentation, and it was great.
Thank you for your invitation.
Anytime. And now, Dr. Vijay, my dear friend. Okay, so the floor is yours.
Thank you very much. Let me share my screen.
You know what I should have done? I should have given you all a random word as well for that thing.
We knew it already, so we did not highlight this discussion.
Too late.
So please let me know if you can see my screen.
I do.
But it's the speaker's version, Vijay.
Oh yeah, it's the.
Wait 1 second, please. I will change. Let me do it again. Okay. First of all, I would like to. Yeah, I'm sharing, so it will appear soon. I hope.
Okay.
It should be there now. Please let me know if you can still see it normal or something changes.
Yeah.
Great.
Yeah. Okay. Yeah. Thank you very much. So first of all, I would like to thank Vladimir naturally for very nice, very kind invitation, and naturally all the organizers, other people, and the university involved. And second, I would like to thank Vladimir again for his very high energy. I think I get motivation out of it. So thank you for that. And naturally, I would like to thank you, both the speakers before me. So they introduced very nicely the abiotic stresses and biotic stresses and what people are suffering with and what is needed to be done. So it makes my job easier, so I can easily go without putting too much emphasis on introduction and more talking about the important points in this area. So in my presentation, let's start with a long time back when humans first time cultivated wild plants and in a way tried to find alternate sources of food. So a very long time. There are evidence existing roughly from 12,000 years ago, and we know a certain area, this Fertile Crescent in Mesopotamia, where it all started. However, if you talk about farming as a system, so you can see that in several countries, at least 7 different locations worldwide where this has been invented independently. What is surprising is that more or less 11 or 12 different plants like corn, rice, wheat, potatoes, or cassava, sorghum, or different type of millets, beans, barley, or rye and oat. So these are the common plants that we all know, and these are the first cultivated crops. However, we marvel at this endeavor, what people did so long back, that it was a revolutionary invention, what they could do years back. However, what is surprising that we are still dependent on those 11 first cultivated plants, and the food comes from at least 90%, 93% or something in this range. Food that we consume today still comes from these cultivated plants. And that may be the first problem where the food security questions start. If we look at current data sets, we can see from, so this is data from World Food Program from 2019, and we can see that roughly 821 million people worldwide have not enough food to eat. And if we go further 2 years later in 2023, or data from 2022, this situation is not getting any better. So it's more than 900 million people at this moment, and mostly in countries where the resources are limited, population density is high, and there is a very, very strong pressure on agriculture. So that's why we think about agricultural sustainability, especially with all the problems that agriculture is facing. When we talk about problems for agriculture or for food security, naturally everyone knows about the weather pattern that is changing. Monica discussed very nicely the problem of salinity, and one major problem of natural contamination wherever it was in groundwater of salinity was one thing, but now with the increasing areas, increasing water rights in the coastal regions, the freshwater areas are getting contaminated more and more with the saline water, which is making it much harder to get enough irrigation water. Naturally, agriculture as a process itself contributes to global warming, which does not help. And then we come to soil quality, and soil quality could also mean the natural presence or availability, which is scanty, patchy in different areas of nutrients, as well as you can think about pollution. So pollution from man-made sources, or even, for example, heavy metals from natural sources. If I give you one example, a very simple example, so study done over 50 years from 1960 to 2009, and what they found out that during this 50-year time period, so you can see it between this graph and this graph, that there is an increase in yield in several areas, and they wanted to find out in this study what are the main driving forces for this increase in yield. And what we find out in this, that if you convert this yield into nitrogen use, or find parallels with nitrogen use efficiency, you can see that all the areas where you have a natural biological way of nitrogen availability, like using microbes, like using symbiotic organisms, there is a very positive correlation existing in terms of gain in yield. However, when we have fertilizers used in most of the areas where it's needed naturally, the soil is lacking nitrogen, and when we use fertilizer, the nitrogen use efficiency does not increase, does not change, and this is a very negative, or even a very poor correlation that exists between increase in yield and availability, that we think we are giving plants nitrogen, but they are not able to use it. And on top of that, it's not only the fertilizer that we are adding and plant can't use it, but this fertilizer, this excess fertilizer is adding up to the soil pollution and groundwater pollution. And that's the whole scenario. That's the scenario around that we are trying to solve a problem. We are trying to solve a problem of less of a certain nutrient, and we are creating two different problems. We are creating a problem that we are using agricultural land which is not giving us the output we want, and the second, we are leading to pollution. So we ask a question, okay, what can molecular biologists contribute? What can they do? Naturally, genetic engineering as a modern solution is available. One can use it, okay, one can think about all the regulations world over in different countries. But because policies are made by discussion, so there is a possibility to discussing. But as a biologist, the major primary question is, what are the targets of these genetic engineering approaches? What should be targeted? So we are talking about nitrogen. So let's give you an example of nitrogen. So for example, if you think in Arabidopsis thaliana, for those who do not know, Arabidopsis thaliana is a very common lab plant. It's not a very important crop plant, but a model organism for all the laboratory work that has been done in plants for last 30, 40, 50 years. And if you think in Arabidopsis thaliana, there are roughly 70 putative transporters for nitrogen, for both nitrate and ammonium. And out of this, only 20% of these 70 putative transporters have been characterized for their what type of regulation is there and what is the functional specificity for these transporters. And now think how much information is missing, how many other things that we do not know about. So we do not know about all the biological processes that are happening and under what conditions. And further on, as I said, Arabidopsis thaliana is not a crop plant. When we go into crop plants, the number of genes only become larger. And because of this, it becomes even more complex situation in the crop plants to understand. And that's why the necessity for plant biologists, necessity is to find novel gene functions or resolve protein-protein interaction networks to actually find out what is a good target for genetic engineering and what is not a good target for genetic engineering. And for finding out these novel gene functions and to resolve these complex networks, naturally we need new experimental approaches where we can actually understand why this natural diversity in genes is existing in first place, why this natural molecular diversity exists, and under what different type of natural heterogeneous conditions different networks are activated. So and that's why it makes sense to have plants tested in an environment of complex combination of stresses than simpler ones. So naturally, it becomes complex to understand, but it's a need of the hour. So if I show you an overview of what is the natural situation with stresses, so you can read here all these names. So if I give you 2 seconds, you can still read 15 names. And all these names that you read are physical and biological factors that in general affect plant growth. Some of them might be helpful under optimal conditions, but some of them are very harmful under different situations. And in nature, all of these factors exist at the same time. Possibly not all of them in situations where they are not helpful for the plant, but in some cases, there might be combination of 1 or 2 or 3 or 4 or 5 or even more. So that's why it becomes important to, number one, identify what are the naturally existing combination of stresses, and then to replicate these stresses in lab under different type of regimes, and then try to understand what and how plants respond. So in this direction, our lab here in Bielefeld, Germany, we are interested in these plant combinations. So I will give you example of 4 different combinations that we have studied in last years, and in brief tell you about what we have learned about them. So for example, first of all, I wrote here arsenic and hypoxia exposure. So you think about arsenic. So if some of you might know that arsenic was an issue in baby products. So all the things that are generated from rice-based food. So all the rice-based food. And rice crops, because they need a lot of water for growth, the hypoxic conditions are very natural to exist in nature for rice type of plants. And for studying, so, and that's why it becomes even more relevant that a natural contamination of soil or of groundwater with arsenic, and a specific type of culture condition for a crop, or even a natural calamity like flooding or excess rainfall, they lead to hypoxic condition. For people who do not know hypoxia, it's a state when plants do not have, or plants roots or whole plant does not have enough oxygen to breathe. Because you all know oxygen amount in the air is plenty. However, the dissolving capacity of oxygen in water is limited. And that's why when you have flooded condition, you have lack of oxygen. And we all know oxygen is very, very, very important, needed for generation, breaking down of sugars and generation of energy. And we all can imagine what would happen to a plant if it can't generate enough energy to fight off such stresses. So what we could learn out of such a combination, that we could find out very specific transcriptional responses and find out more than 800 specific arsenic and hypoxia combination responding genes. And naturally, the next target is to find out meaning into what these transcriptional changes mean. If I give you another example, we all have hundreds of different kind of electronic devices at home. I mean, I don't want to name them because it's impossible to name them, because now everything is electronic, from your toothbrush to your watch to your phone to your, I don't know what else. And these electromagnetic waves that these all electronic devices generate next to a biological system that you are, and as well as all other life forms, including plants, are harmful in different ways. So in this setup, we try to understand how a combination of different intensities of EM waves can affect plant growth and can affect important biological processes, for example, photosynthesis. And in this case, we could find out that at least for photosynthesis, that the combination that we have used in this case could directly affect or had indirect effects on the pool sizes of important complexes from photosynthetic electron transport chain, like plastocyanins or photosystem 1 or ferredoxin pools, as well as photosynthetic yield of the plant. So naturally, it becomes an area of concern, and one should further understand why these interruptions, or why these indirect or direct effects of these EM waves happen on the plant. If I look into another such combination, Monica introduced salinity. So I will just add that heat and highlight are part of the whole process, because you know plant needs light to grow, right? They have to do photosynthesis, so they need light. And in areas that are with very high temperature, so for example, most of India, which is suffering from temperature like 45 degrees or 48 degrees, or even Brazil. So Vladimir just told us today that it is a whole year summer now. There is no real winter. So heat, excess sunlight, that also brings highlight into this equation. And at the same time, if you have salinity or saline soils where these plants are growing, this type combination is a very naturally existing combination. And to understand it, it's very important. And we could understand in this very complex combination of 3 different stresses, we could find out that salinity exposure still dominates very strongly. I have to mention that this was a study done in a plant which is significantly tolerant to salinity compared to all other plants that are known for its sugar beet, and sugar beet can tolerate very, very high saline, even like seawater saline conditions. And still, we could find out that salinity has strongest effect out of this combination. And further on, I will go one further to a combination of ROT, which is part of heat equation, and then pathogens like Pseudomonas. Pseudomonas is a natural pathogen in, for example, different crops, potato, tomato, you name it. Several Brassicaceae members like mustard. So in this combination, we could find out new elements in salicylic acid, jasmonic acid, and abscisic acid signaling networks. So why I'm mentioning these three important hormones, very important to say that these three hormones are necessary for plant acclimation to pathogen stress as well as drought stress. And that's why we could find new elements through this combination in the interaction network or signaling network of these hormones. I will go in a little bit more detail into arsenic and hypoxia exposure. So among these 800 something arsenic hypoxia responsive genes, so I will give you a little bit detail into this. So the experimental setup was a hydroculture. Monica mentioned very detailed what is a hydroculture and why it is needed and why it is important. So in this case, we did a hydroculture. In a hydroculture, it's very easy to manage or maintain a specific arsenate amount in the growing media, growth media, as well as how much oxygen is inside. So in this setup, we could find out that there is a very, very nice interaction of hypoxia and arsenic happening. So arsenic, very briefly, is if you look at the atomic structure, it's an chemical analog of phosphate. So phosphate is an important nutrient for plant, and plant can't distinguish sometimes between arsenate and phosphate, and arsenate is uptaken. However, when arsenate is inside the cell, plant knows, oh, I can't use arsenate, I need phosphate. So what they try to do is they try to suppress a specific type of transporters for phosphate, and some other transporters are still functional, so they can uptake uptake phosphate, but suppress phosphate uptake. And this generates a specific kind of phosphate starvation response, because now you are lacking phosphate, but you have enough, a lot of arsenate. And very interesting, what we found out that hypoxia independently also generates a very similar kind of, or very similar in terms of that it's a phosphate starvation response, but different that the molecular makeup of the network that is highlighted is different. So that is activated as different in the plant. So why this happens? So hypoxia in general leads to lack of nutrient uptake. So it suppresses nutrient uptake because, as I told you before, plants have less energy now, and this could lead to lack of nutrients, and this generates naturally a phosphate starvation response. But what was interesting to find out in this study, that when we have this combination of arsenic and hypoxia, the combination behaves like hypoxia. It does not behave like arsenic stress. It behaves like a hypoxia stress. So hypoxia dependent phosphate starvation response is more dominating in this case. And the response potentially tries to lead towards replacing phospholipids from the membranes to galactolipids. So this phosphate can be used for other processes that are more important. As I said, we had roughly more than 800 genes that were specific for this combination. And out of these genes, a chunk of these genes, we could find out that they are important, or they are needed for the plant for important root modifications. And what are these root modifications? These are root modifications called root hairs. So just briefly, I have to tell that root hairs are modifications of epidermal plasma membrane. So root hairs are not, so there is no need for new cell division. There is no need for cell division and growth. So there is just a need for pushing a membrane to elongate in form of a hair. So plants have this adaptation under this specific combination, that they can generate root hairs instead of the actual roots, or excess root branching because they don't have enough energy. So what they do with their limited amount of energy, they invest it in direction of generation of root hairs. So we saw, maybe it's much better with a picture, so I can show you here. So you can see with the top two, so this is our control experiment. So naturally root hairs are common in all roots. So as you can see, there are root hairs. For arsenate, you also see a certain amount of root hairs, but as you can see at the bottom, for hypoxia and the combination shows a very, very strong increase in root hair growth. So I tried to summarize this study with 6 different points. So what we saw very early, that we saw a very strong redox dependent change in the root cells. And then we saw phosphate starvation response, as we saw, as I told you before. And now plant has to decide, okay, how much energy do we have? How much energy is required for this process and that process? And how much of energy do I have? And how much energy I can use for growth? And how much energy I need for defense? And that's why they look into phosphate availability. They have to increase, they want nutrients, and that's why they increase the root hair growth and then generate more root hairs. And at the same time, they suppress the apical meristem, but they maintain the identity of the apical meristem. So later on, when more air is available, when re-aeration happens, when the flooding water has receded, they can still grow. So I hope I am able to go through these findings and highlight that there is an urgent need for this type of combinations to be understood, to be identified in nature, and then studied in laboratory environments, and to understand, to help for genetic approaches to find to fight for agriculture sustainability. So I would like to end on a positive note. The problem with all of us, problem with in general with humanity, is that we are too focused maybe on our limitations. So I give you these 2 photographs. If you look at this, you see, okay, there is some plants growing, but you don't see anything special here. You can't identify these plants. But you look on the right hand side, and you can see, oh wow, nice, it's maize, and you see nice maize plants growing. However, if I tell you that before domestication, so the maize plant was this. So humans domesticated this plant and made it into this one. Over 9,000 years of change, this plant is unidentifiable. You can't say in nature that this plant is this plant, but it's the same. So maybe as human beings, we have to look at what is possible for us to do. What are the positive things? What are the things that we can actually make difference in, and then strive for those. With these words, I would like to thank all the collaboration partners for all these results and studies that I showed you, and naturally the lab members, Jana and Madita, who are here, and thanks again to Vladimir and all of you. Thank you very much.
Thanks, Vijay. Thanks a lot. Where is that last picture from? Just out of curiosity.
That's Shimla. Sorry, that's Shimla. That's my, let's say, alma mater. That's where I studied.
All right, amazing.
That's in Himalayan region in India.
Northern India, right? Amazing. I think it ends on a very high note. Your presentation, when you stressed so much the need for agricultural sustainability. That's what we wanted to talk in this webinar, to highlight that it is possible to have agriculture with sustainability, aligned with sustainability, especially because agriculture and virtually any other human activity depends on ecosystem services. It depends on natural resources that and processes that need to be maintained and carefully used. Otherwise, the whole concept of sustainability goes away, right? And I know it's difficult for us as a species, as humans, to remember that we have a responsibility not only for the present, but also for the future. We are a species very limited in remembering that the planet will be here for other people after us, that we have a certain time on this planet. It's also interesting when we talked about this combination of stresses, of naturally existing conditions. I also took a picture of the word cloud you presented there about the different kinds of stresses plants are subjected to. And I think these approach, when we have to address this combination of stresses, is especially important in view of a changing climate, which is what we have everywhere, and a somewhat unpredictable climate. We don't know what will happen. We don't know how things will become. Here in Brazil, in the semi-arid region of Brazil, we have talked. I mean, this is a semi-arid region, and the climate was always seen as an enemy. And it's a region that was historically neglected for centuries, and only relatively recently have we started to acknowledge that the climate is often not an enemy. We have to know how to coexist with the conditions and how to take advantage of what we can without degrading even more the situation. So I think it's really amazing what all we have seen here today. I think Monica has a comment.
Yes. Hi, Vijay. It was a very nice presentation. Indeed.
Thank you.
Yeah. One of my favorite areas, heavy metal stress, and that too arsenic. So, okay. Few technical questions just to for my some experiments clarity. So, yeah, arsenic you have given in +3 or +5 form the stress.
It's arsenate, Monica.
It's arsenate, so that is okay.
Yes.
Then what concentration?
250 micromolar.
250. That means 25 millimolar, right?
No, 250 micromolar. Sorry.
Micromolar, right?
250 micromolar. 25 millimolar. I don't think that Arabidopsis can survive. Maybe Teres vittata can, but I don't. Teres vittata is a fern which is tolerant, so they can survive, but not Arabidopsis.
Okay. One interesting thing I've seen is the transcripts that you have retrieved. That data analysis that you would be doing. So there was some phosphate starve response transcript as well, right?
Yes, indeed. I mean that's what I wanted to highlight. Yes.
Yeah. So it's kind of nutrients. I'm not able to correlate because it's kind of nutrient stress, right? So
yes, please.
Have you set up the experiment like individual arsenic stress, individual hypoxic condition, combined arsenic hypoxic, and are you getting this phosphate starve response transcript in common in all 3 individual experiments, or you have got only while giving the combined stress?
Yeah. Okay. Let me explain. So this is very important. So what is known for arsenate toxicity, as I said before, arsenate as a molecule, as an atom, is very similar in structure, in size to phosphate. So plants instead of taking phosphate accidentally take up arsenate, and they realize only when arsenate is in cytosol, or they have to compartmentalize it into vacuole, and that's when they realize and they decide, okay, we have to suppress these specific type of transporters that are uptaking phosphate, so we suppress them. And as soon as you suppress phosphate uptake transporters, naturally, on the other hand side, there is a lack of phosphate as well. So that generates phosphate starvation response.
Did you able to retrieve any novel transporter for arsenic hypoxic combined stress condition?
Yeah, so we have roughly 800 of them. So yeah, we are going through them and trying to find out individual.
See, as far as arsenic, heavy metal, I know it does not have its own dedicated transporter. It always mimics some of the other ions.
Naturally, we don't find the transporters for arsenate, but we find several transcription factors, for example, that are important for regulation of the uptake.
Okay. 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 I'll be having more questions. I'll be asking you in person, Vijay.
Yes, please, you can text me.
Yeah. Yeah.
Oh, feel free if you have any questions here. We still have a little bit of time. It's not a problem. So does anybody in the audience have any questions? Vijay? Yes, we do have a question in the chat. Leticia, she asks, hi, I would like to know about what would be more efficient and possible in a world with climate change, adapting species to survive, as you have shown with maize, or changing the way we deal with agriculture?
So, I mean, if I want to, so it's a broad question, let's say. So if I answer it in a very simpler term, so first thing I would say is definitely agriculture has to adapt. So there are several regions where you can't do agriculture anymore because the soil is already polluted, salinated, heavy metal, whatever contamination. So or even in some areas there is not enough water. So naturally, either we have to find a source of water for these places, find a source of removing the contamination, or the agriculture has to be moved to a place where you can live. And that's why the agroforestry example that we are talking about, or pest control, if the pest is a problem for a certain area, those are changes. But I think one thing that I would definitely emphasize on is that, so if you can see in last year, so I know it from my village somewhere in India. So we can see it. I can tell you the temperatures from last 10 years, that we see a shift of all all seasons little bit further. So some seasons have grown. So all seasons have, let's say, moved from their original position. So maybe slowly, at least in over a decade, we have to change how we were growing crops before. So that's an adaptation that can be made easily, because this is something that this data exists already. We know that this data exists and this won't change in a short term. So over the next 10 years, even if we don't have very resistant varieties, we can definitely slightly change it. So I was having a discussion with my father yesterday, and he mentioned that now people in the village have gone away from potato cultivation because potato was suffering more, and they have gone towards other crops like mostly vegetables, and they are still earning as good as they were earning before with potato. And that's what I mean. It's a very smart idea to do it through this way, and it would be very different from one place to other place naturally. So it will vary from, you have to understand the natural local conditions, and you have to see what is changing with time, and then you have to deal with it.
Interesting.
Thank you for question.
Okay, we have Monica and Yana also with questions.
Vijay, you have performed the experiment with A3 till which stage? I mean, vegetative, productive, or like?
Can you say these were adult plants, Monica? These were in the 5th week of their growth. Adult plants.
Have you tried working with some other, giving this combination of stress to other cereals or legumes which are heavily impacted with these heavy metal stresses?
I think the best would be rice actually, but the problem with rice is that rice is very, let's say, hypoxia tolerant. So you would not see very much of a combination, but just an arsenate effect. So that's why we went with Arabidopsis, because we want to understand what processes are actually where the interaction is happening, and that's why we chose Arabidopsis.
Okay.
I think Yana has a question.
It's not really a question, it's more a comment to Vijay's answer of the question. So I totally agree with Vijay, and I think it's kind of not a decision by human beings to change their agriculture system, but it's more they are forced to do it. So I would like to give an example. My father had some agricultural land. So he's not doing it by himself, it's just another guy. And he told us this year, we wanted to bring out maize as typical here in April or May, maybe. and they told us, he told me like, okay, it was just not possible because the soil was so wet because there was so much rain in Germany that they just couldn't enter the field, so it was not possible. So it's just it just came in my mind that it's maybe not about a really decision, but it's more like, okay, you can decide it now, because when you decide now, okay, you just change our system, it's fine. But I think in a few years, maybe not a few, but in a decade of years, humans are just forced to change their system, because otherwise it will not work out anymore.
Yes.
There
Well,
was
I think we see that everywhere, right? I mean, sooner
Yeah.
or later we will have to adapt. Otherwise, we can face the consequences. And it's much more traumatic and much more expensive if we choose the latter option.
Yeah, especially in terms of climate change. I don't know if you know about the big floods in southern Germany at the moment.
I have heard about it.
So I'm pretty sure there are a lot of videos online about Bavaria. So it's flooded everywhere. So even if they had the chance to bring out their plants for agriculture proposed, they will just lose it now. So you know what I mean? So it's this whole complex situation that we have to face.
Yes, we are facing big floods in southern Brazil as well. And southern Brazil is responsible for nearly 90%, if I'm not wrong, of rice production in the country. I mean, Brazil is a self-sustainable country with regard to rice production, its consumption. But because of these floods in southern Brazil, the country has had to import rice from other neighboring countries. And I mean, these impacts we are seeing in southern Brazil, it's also because we have failed somehow to adapt to a change in climate. And I mean, a lot of it depends, of course, on political measures, how to address these challenges. And what we have seen here, unfortunately, is a choice about not addressing these problems. And of course, the bill has come, the consequences are here. And I mean, we will have to adapt at some point because it's a situation that cannot be sustained. So yes, in agriculture and in every other field. You guys have any other comments? Or, I mean, we are approaching our ending time. We only have 8 minutes left, actually. So if you would have any closing remarks, that would be the time.
Yeah, I would like to give a last comment. So on what you just said. So you're talking about Brazil, but as far as I know, at least 3 countries, so at least people out of 3 countries are in this meeting now. And I think it's everywhere. So it's not a Brazil problem, it's an overall problem, and we all have to face this. And yeah, we can spend time on research to solve this problem. And I'm pretty sure a lot of students maybe decide for this kind of subject because of this reason. So I just want to let you know there's a lot of research to do, and we should all face this problem. And yeah, it's a good field to study, and just stay motivated, even if it's hard time and if it's not only positive words in here, also here in this webinar.
Yes, I mean, we are preaching about the end of the world, but also how to deal with it.
Yeah,
Yeah,
I would like to. Yeah, okay. Yeah, Vijay.
Please go on, Monica.
Yeah, I would like to end up this webinar saying that we need to outreach. We need to develop awareness programs. We need to talk to our ancestors, those who were not educated, but they were practical hands-on experience. We need to go to them, as Vijay had shown that figure, and Jana also mentioned about his father. So they are the real, players who are every second they are working in the field. So the challenges for us, those who are doing at the lab level, talk to them, ask them what you are noticing in general over a century. I think they would be like, more, because yes, whatever we can do, we are doing at lab level. We need to transfer the technology. So for that, we need to reach to the center of the circle, and which is the farmers, which are the people who are working in this field for since so long. I think morphological interpretation may quickly help us to resolve the problem. So yeah, that much. Yeah, and yes, and field visits should be a mandate in all curriculums. That the students should talk to the local people residing over there, that how they are handling all such kinds of thing, how they are maintaining their food security things. So yes, that way. Yeah, thank you.
Vijay, you have any comments?
Yeah, no, so I just wanted to say that in general, we are talking about very, big topics, big problems. And what I would like to, at least for students who are upcoming and are new with this and want to pick up or not pick up research and think about, so it's not about how big a difference you can make. It's not about if you can bring out a revolution, but I think every tiny thing, every small thing, every person's mind who is interested in this and wants to contribute, I think every single small idea is helpful. So you just have to find, I mean, I'm very much thankful to Vladimir. I think these are things very important to discuss, naturally, hard to understand, very hard to make conclusive arguments on, but definitely important to discuss and to let's say have a motivation forum for all the young people, so they can take up research and bring out new ideas. Yeah, thank you again.
Thanks, Vijay. Thanks, Yana. Thanks, Monica. Madita as well, who has been there patiently. If you want to say anything, of course, you're totally welcome.
Somebody has a question in the chat, I think.
I think she does. Larissa, would you like to say something? I don't know.
Yeah.
I think not. Okay. So, okay, guys, it's been really an honor to have you here. Again, I hope this meeting can happen in person someday soon. And thank you not only for being here, but as Vijay said, and I totally agree, this is a very interesting moment. There's a lot of students in this meeting, and of course, surely many of them will be inspired by what they have heard and seen here today. And this is an interesting platform, so we can interact, students can interact with our speakers and vice versa. So should have any other questions or would like to contact any of our speakers, you could write us an email and we can make this contact bridge. So guys, thank you again. I really appreciate you spending your time. I know it's a bit late in India and not so much late in Germany, but anyway, I thank the effort. And thank you all. I think that's it for today. We are ending this session. Thank you again, and we are here anytime.
Yeah, and thank you to Vladimir again.
Okay. See you guys. See you soon. It was an honor.
Yeah, good luck, everyone. Bye-bye.
Right. Bye.
Bye-bye.
Bye.